This is lecture preview number 2 coming to you guys. We will finish up the last portion of this lecture. And we will begin here with contraction and relaxation of the skeletal muscle. So, we're going to talk about the contraction cycle that has to occur. And I discussed with you guys in class, and for those of you watching online, about rigor mortis and how rigor mortis is a condition that occurs after humans have died. And this is due to sustained contraction. This is stiffening of the body that happens. So this is due to deficient ATP production. So what happens is you have hydrolysis of ATP, and this is just a fancy way of saying that ATP is split apart. So I want you to know these 4 steps. Again, I said that these were important in lecture, that they're testable. So ATP is split apart, and you have something called the cross bridge formation. And this is just, again, this sliding filament model in which myosin and actin are attaching to each other. So after you have attachment of myosin and actin, you have something called the power stroke, and this is where the myosin and actin cross bridge rotates, and sliding of the filaments actually occurs. And then eventually we have detachment of myosin from actin, and this is where muscle relaxation occurs. So in order for myosin to detach from actin, ATP has to be present. Okay. So as the next ATP binds to the myosin head detaches from actin. So ATP will bind to myosin, and then myosin will detach from actin. So just go through this again. Again, these 4 steps are important. You have the splitting of ATP through ATP hydrolysis. You have the formation of cross bridges. You're going to have the power stroke, which again is just going to be where actually the filaments slide past each other. And then you're going to have detachment of myosin from actin. And we know that myosin must bind to ATP in order for relaxation to occur. So this is important. The contraction cycle repeats as long as ATP is available and there is sufficient levels of calcium. Remember, I said that calcium needs to be present in order for muscular contraction to occur. This is just a picture of showing you the relaxed muscle, how the muscle is relaxed. And as the muscle shortens, it gets closer. Z disc and Z disc get closer together. Here you can see the Z disc. Here you can see the Z disc. You can see the muscle is shortening. Again, this picture here will show you fully what's happened. Myosin head is hydrolyzing ATP and splitting it. As you can see, as it begins to split, myosin will then bind to actin, forming the cross bridge, and then during the power stroke, they will slide past each other. Okay, and then eventually, whenever ATP comes in, it will then bind to myosin, allowing detachment from actin. There is this again is just talking about excitation contraction coupling. So, an increase in calcium in the muscle starts muscular contraction. That's important to know, that the increase in calcium concentration causes muscular contraction, right? So as calcium moves in, it moves tropomyosin away from the myosin binding site. So I want you to know that calcium moves tropomyosin away from the myosin binding site. So tropomyosin it's very similar to myosin, but just slightly different, okay? Tropomyosin. So calcium moves this tropomyosin away from the myosin binding site, and allows the actin cross bridge to form. Okay. So calcium moves tropomyosin away from the myosin binding site and allows myosin to bind to actin, allowing for the cross bridge to form. Here is a picture showing you relaxation and contraction. During relaxation, troponin holds tropomyosin in position to block the myosin binding site, right? We don't want myosin to bind to actin here during relaxation. We're wanting the muscle to relax, so troponin is blocking the myosin binding site. During contraction, calcium binds to troponin, which changes the shape of the troponin tropomyosin complex, and it uncovers the myosin binding site on actin. So that's during contraction. So these words I said was important, putting specific emphasis on synapse, neuromuscular junction, and neurotransmitter. I told you that in class I showed a good example of Carson and Gavin. I said that Carson was a somatic motor neuron, and there would be input to the somatic motor neuron, and then the neurotransmitter would be released and eventually go across to the neuromuscular junction and bind to the motor end plate. So here we'll talk about these words in detail. The motor neurons have thread-like axons, and axons just carry action potentials. So this either extends from the brain or the spinal cord to a group of muscle fibers. Okay, so action potentials arise at the interface of the motor neuron and the muscle fiber. So this is the neuromuscular junction. So this space in between the motor neuron and the neuromuscular junction, this is called the synapse. So this is where communication occurs between the motor neuron and the muscle fiber. This is called the synapse. It's just the space. Synaptic cleft, I'm not too worried about. It's just a gap that separates the two cells. The neurotransmitter, this is important. Chemicals released by the initial cell communicating with the second cell. So the somatic motor neuron is going to release a neurotransmitter, which is then going to go to the muscle fiber or the motor end plate. So this is going to be the region of the muscle cell membrane opposite of the synaptic end bulbs. The motor end plate is going to contain acetylcholine receptors. And again, we're going to talk about how acetylcholine is primarily going to be the neurotransmitter involved with muscular contraction. So nerve impulses elicit a muscle action potential. So this action potential is just going to be this electrical signal that occurs. So there's going to be 4 different steps, and all these steps are going to be important for examination purposes. There is a release of acetylcholine. So nerve impulses arise at the synaptic end bulb, causing many synaptic vesicles to release acetylcholine. So I just want you to know there's a release of acetylcholine. Acetylcholine has to bind to the acetylcholine receptor. And after this produces a muscle action potential. Okay, there's going to be the inflow of ions. Sodium is going to move inside the muscle fiber and make it more positively charged, which is then going to cause depolarization and allow the action potential to travel down the axon. And I want you also to know the termination of acetylcholine activity. This occurs through the enzyme acetylcholinesterase, right? Acetylcholinesterase is going to terminate the activity of acetylcholine, breaks it down. So just to go through this again, you're going to have the release of acetylcholine. Acetylcholine is going to bind to the acetylcholine receptor. From there, this is going to generate a muscle action potential. And from the muscle action potential, it's going to be terminated by acetylcholinesterase. This picture right here will not work due to animation being present. This is just showing you what I've already described to you in detail. Acetylcholine is released from the synaptic vesicle. This is occurring here. Acetylcholine is being released. This is the synaptic space, and it's moving over here, and it's trying to bind to the acetylcholine receptor. Right? As you can see, the muscle action potential is generated because acetylcholine is bound to the acetylcholine receptor. Depolarization is occurring as sodium moves in. So muscle action potential is produced. And then there's going to be acetylcholinesterase, which is going to be present, and acetylcholine will be broken down. So we talked about botulinum toxin, and I wanted you to know that botulinum toxin is going to block the release of acetylcholine from the synaptic vesicle. Okay, so botulinum toxin blocks the release of acetylcholine from the synaptic vesicle. And I talked to you guys about where botulinum toxin was produced, how it came. It came from a bacteria called Clostridium botulinum, and this bacteria was going to be present in improperly canned foods. It's also present in spore form in honey. And babies aren't supposed to eat honey less than a year old because they can develop something called floppy baby syndrome, right? In which they become paralyzed due to the blocking of acetylcholine from the presynaptic vesicle. So this is also a cosmetic treatment to relax muscles that cause facial wrinkles, and I think that's kind of cool. So it's very hard for the for a contraction of the muscle to occur. So it's relaxation of facial muscles. So I said that this would definitely be exam worthy. This will show up on your exam. The mechanism of action as well as its medicinal uses. What's important on this is going to be anticholinesterase. Um, Actually, no, don't worry about these two. I'll talk about them briefly. Curare is going to be a plant poison in South America. It's going to cause muscle paralysis by blocking acetylcholine receptors. Rather, botulinum toxin. So, muscle fibers have three ways to produce ATP from creatine phosphate. Again, if you are familiar with creatine um, supplements, this is a way for uh, uh, our muscles to generate ATP. ATP is needed for work. It's the energy currency of the cell. So by anaerobic cellular respiration, and I said this was important because it doesn't require oxygen. It's again going to yield um, fewer ATP molecules, and by aerobic cellular respiration, and this requires oxygen, and thus will generate more ATP production. So, a huge amount of ATP is needed to power the contraction cycle. Again, lack of ATP, there's going to be no contraction that occurs, and your body is just going to stay in constant tetany, also known as rigor mortis, and to pump calcium into the sarcoplasmic reticulum. Right, sarcoplasmic reticulum is going to store calcium for calcium to be released, bind to troponin, and cause it conformational change in the troponin tropomyosin complex. Um, Here, ATP is being generated from creatine phosphate. As ATP is hydrolyzed into ADP, we see here that creatine is becoming creatine phosphate and allowing muscular contraction to occur. And then we see that muscle glycogen. This occurs through a process called glycogenolysis, in which glycogen is broken down into glucose. Glucose can then undergo into the glycolytic cycle and then produce pyruvic acid. Pyruvic acid can be broken down into something called lactic acid, and lactic acid will often occur in these fast twitch muscle fibers um, that are going to have some anaerobic activity. Okay. Again, this here is cellular respiration, which occurs in the mitochondria. If you took me for Biology 112, we discussed this in detail. We talked about the many enzymes that are occurring um, within cellular respiration. So, oxygen from hemoglobin and myoglobin. I said that those were going to be the two resources for oxygen, right? Hemoglobin and myoglobin, right? Oxygen comes from hemoglobin and myoglobin, right? It's a final electron acceptor within the electron transport chain, and it's coming from hemoglobin and myoglobin. I think that's important to know. So, metabolism, anaerobic respiration. I said that the important thing to know is a series of ATP producing reactions that do not require oxygen. They don't require oxygen. Okay, we said here that glycolysis breaks down glucose into molecules of pyruvic acid and produces two molecules of ATP. Right? If oxygen levels are low, right? If oxygen levels are low, anaerobic reactions convert pyruvic acid to lactic acid, which is carried away by the blood. I want you to know that this lactic acid production occurs in the absence of oxygen. Here we talked about aerobic respiration, and I said that um, aerobic respiration would require oxygen. So this pyruvic acid is able to, instead of being converted into lactic acid, it's able to enter into the mitochondria to be completely oxidized, thus generating ATP, carbon dioxide, water, and heat. So we need to know that the muscle tissue has two sources of oxygen: hemoglobin in the blood and myoglobin in the muscle cell, and that's important to know. Motor units we talked briefly about, but I said it again that I probably wouldn't test you about these, but I talked about this in terms of what consists of a motor unit. So motor units is just going to be the motor neuron and the muscle fiber it stimulates, right? This is all going to be a unit itself. So the axon of the motor neuron branches out, forming different neuromuscular junctions with different muscle fibers. This makes sense. Controlled precise movements consists of many small motor units. Voice production has 2 to 3. Eye movements has 10 to 20. Muscles in the arms has more. So, as the muscle continues to grow in size during, if you're working out and hypertrophy occurs, you're going to increase the amount of motor units, thus increasing the number of axons and neuromuscular junctions. This is an example of a motor unit. This is the spinal cord, which is projecting these motor neurons out, and they're going to different muscle fibers here. So again, the motor neuron and the muscle fiber itself makes up a motor unit. Twitch contractions, the brief contraction of muscle fibers in a motor unit in response to an action potential. So an action potential comes in, and thus we have a twitch. There's a period called the latent period and the contraction period. During the contraction period, this is when calcium binds to troponin, right? This is when myosin binding sites on actin are exposed and the cross bridge forms. That's awesome. So once after we have contraction, we have relaxation. Relaxation is when the myosin binding sites are covered by tropomyosin, right? Tropomyosin is bound to myosin, thus myosin can't bind to actin. So I would like for you guys to know that the contraction period occurs, then you have the relaxation period, and the latent period is the period in which no muscular contraction can occur. So again, just to go over this, the contraction period is whenever calcium binds to troponin, right? Myosin binds to actin, the cross bridge forms. That's what I want you to know for the contraction period. The latent period is going to be a period where there is a delay in the stimulus reaching the motor unit and the muscular contraction. So in the relaxation period is a period in which myosin head detaches from actin. So isotonic and isometric contractions. Isotonic contractions, we said these are going to be used for situations in which you're picking up a book off of a table. And I told you that the tension remains constant. So there's constant tension in the muscle, but the muscle changes its length, right? So an isotonic, iso means same, so same tone, right? You're having constant tension, but the muscle is changing in length. Notice how this differs from isometric. So during isometric contractions, right, the tension generated is not enough for the object to be moved, so the muscle does not change in length. Okay, this is holding a book steady using an outstretched arm. So if you stretch out your arm, this is an isometric contraction, right? There is no change in length of the muscle, but during an isotonic contraction, there is constant tension changing the length of the muscle. This is picking a book up off the table, and both of those things are important. This is just a picture here kind of demonstrating that for you. No movement here. This is an example of an isometric contraction. But as he's picking the book up, this is an isotonic contraction, as there's a concentric and an eccentric movement. Red muscle fibers. There's two different types of muscle fibers, right? We talked about the motor unit and how the motor unit would be made up of the somatic motor neuron and it would go to the muscle fiber, right? There's two different types of muscle fibers. There's red muscle fiber and the white muscle fiber. What makes up or what is the difference in the muscle fibers is the amount of myoglobin. And remember, we said myoglobin was going to be the protein component that was responsible for binding oxygen within the muscle fiber. So hemoglobin does it in the blood, but myoglobin does it within the muscle. So myoglobin is going to be very rich in red muscle fibers. I said Sha'Carri Richardson. Remember the track star from LSU? She has a lot of myoglobin, right? She's going to have more myoglobin than Ricky Joe up the holler, who's drinking 6 packs of Natty Light, right? Because she is an athlete, right? She needs more mitochondria. So she's going to have more mitochondria because more oxygen is going to be required because she has more muscle and she's going to have more muscular function. Whereas white muscle fibers have lower amounts of myoglobin. They're going to appear lighter as well. So there's 3 different types of muscle fibers. They contract at different speeds and vary in how quickly they fatigue. So muscle fibers contract at different speeds and vary in how quickly they fatigue. They're classified into 3 different fast oxidative glycolytic, fast glycolytic fibers. Slow oxidative fibers, these are going to be capable of prolonged, sustained contractions for many hours. I want you to think about marathon runners, right? These are the muscles in the back which are responsible for maintaining posture. So muscles in the back that are responsible for maintaining posture are slow oxidative fibers. Okay? These have slow speeds of contraction. They're very resistant to fatigue. So if you think about it, these muscles are able to have sustained contractions because in order to stay upright, the muscles will constantly have to be able to contract. So these fast oxidative glycolytic fibers or fast glycolytic fibers are going to be responsible for, they're going to be moderately high resistant to fatigue. They can generate some ATP by anaerobic glycolysis. I think that's noteworthy. And the speed of contraction is much faster. So these are going to contribute to activities such as walking and sprinting. So um, fast twitch fibers, these are going to be more prevalent in sprinters, right? Marathon runners are going to have more slow oxidative fibers, whereas sprinters are going to have more fast twitch, fast oxidative fibers. So ratio of fast glycolytic and slow oxidative fibers are genetically determined. Individuals with a higher proportion of fast glycolytic fibers excel in intense activities such as weightlifting and sprinting. Individuals with higher percentages of the slow oxidative fibers excel in endurance activities such as long distance running. So, various types of exercises can induce changes in muscle fibers. Aerobic exercise transforms some fast glycolytic into fast oxidative glycolytic fibers. Endurance exercises do not increase muscle mass. Cardiac muscle tissue. Um, I said that I was going to test you on the same things that I tested you previously. This is the principal tissue in the heart wall. Okay, they have the presence of intercalated disc, which are going to connect the cardiac muscle fibers. So these allow muscle action potentials to transfer from one cardiac muscle to another. Okay, requires a constant supply of oxygen. Okay, it is able to use lactic acid produced by skeletal muscle fibers to make ATP. Again, smooth muscle tissue, usually activated involuntarily. It's not voluntary, right? Action potentials are spread through fibers by gap junctions, right? Fibers are stimulated by certain neurotransmitters, hormones, or autorhythmic signals. This is found in the hollow organs, right? Small intestine, large intestine, duodenum, stomach, things like that, as well as the arteries and veins. I think it's worth noting that these are not arranged in orderly sarcomeres as the skeletal and cardiac muscle are. Okay. And that will primarily conclude our lecture. Thank you so much.